Does that look all right? Um, all right. So, uh, thanks for coming to the
Oops,
talk.
sorry, Kyle, you've got your presenter view um, up instead of the.
Oh, sorry. Is that okay? Or
Yep, that's better.
okay. Um, yeah, thanks for coming to the talk. This is actually work by one graduate student in my lab, Andrew Rubio, a former student in my lab. Andrew Rubio is now at Clemson, and a host of other collaborators is listed here. So, when we talk about deimatic behavior, we're talking about a defensive tactic where an animal suddenly reveals a startling feature like bright colors or large eye spots to frighten or distract a predator, an approaching predator, giving the prey a chance to escape. And we think this South American uh, grasshopper, Paramastax nigra, may engage in this type of display, a deimatic display. And so the work I'll be talking about today is just basically exploring that possibility from a couple of different angles. We studied this species in the foothills of the Andes near Tarapoto in San Martin province in Peru. And so here is the grasshopper in question, and when approached, it will engage in this display where it leaps into the air and unveils these bright white underwings, and basically becomes a kind of flying white blob. Um, so I'll show a couple of videos. Here's a captive grasshopper just revealing the underwings. And then this just shows a kind of a short video of the um, grasshopper leaping into the air when approached and then revealing these underwings and then flying away, um, and ultimately landing on another surface, typically on a leaf. And I'll talk about that in a minute. Oops, sorry. So basically, we're interested in a number of basic questions that we could answer just using observational and minimally manipulative approaches. First, we wanted to ask just the basic question of whether the primary defense for this grasshopper is camouflage, which is kind of a prerequisite for a display being deimatic. I mean, they start these for in deimatism, the animal is supposed to start out trying to be camouflaged against a particular background, and then if that doesn't work, it engages in the secondary defense, a deimatic display. And so our first question was, is the primary defense camouflage? And so you would predict if that's the case, then either the color or the luminance of the grasshopper would match the appropriate background. In this case, the grasshopper is green, so a matched background would be a green leaf. And so to test this, we looked at whether the color or luminance of the grasshoppers that we sampled did indeed match a leaf background compared relative to an unmatched background, which would be just the plain dirt or soil brown background. And then the second test is whether grasshoppers prefer to land on the matching background of the leaf after they are disturbed and engage in this display where they fly up into the air and then move to a different spot. And then in terms of the secondary defense, whether this is a deimatic display, we had several predictions that we thought would be appropriate for investigating whether it's a deimatic display. First of all, we predicted that the grasshoppers would be more likely to initiate the display when they're found on a non-matching or soil background. And so we had a couple of tests looking at this. First, whether the distance at which the grasshoppers initiate the display is greater if they're on an unmatched or soil background. And secondly, whether physical contact was required before display initiation, arguing that it would be they would be less likely to tolerate physical contact if they're on an unmatched background. And then we also looked at the prediction of whether the secondary, presumably deimatic display is energetically costly, which is, again, one of the general features of deimatic displays. And so we looked at whether the distance grasshoppers traveled during the displays would decrease over sequential displays. So for methods, we looked at whether we were working in these old growth forest trails near Tarapoto. We walked trails and recorded individuals either on unmatched or soil backgrounds and on matched or leaf backgrounds. And then we approached the grasshopper until they initiated the display. And then we would follow, as much as possible, we would follow the grasshoppers, and they would then repeat the displays over and over until we lost track of them. So we had sequential displays. And then we measured the distance to the grasshopper before a display was initiated, and then the distance they traveled while doing the display. We recorded the substrate they landed on, and then took photographs of the grasshopper and then the nearest matched and unmatched background. This was done with the Mica toolbox package implemented in ImageJ, and then we quantified color and luminance, the color and luminance match of each grasshopper to leaf and soil background photos using an avian visual model to calculate JNDs, or just noticeable differences. For statistical analyses, in most of the comparisons, we were using linear mixed effects models, or LMMs. But for whether or not the grasshoppers preferred to land on matching backgrounds following the display, we used a general linear mixed model, or GLMMM. And then for the question of whether physical contact before the display was more common when the grasshopper is on a matched background, we used logistic regression. So just a brief overview of the statistical methods. And so on to the results. So we looked at color and luminance matching. We did not find evidence for color matching, comparing the grasshopper color to green leaf versus soil. But we did find luminance matching, where the grasshopper matched the luminance of the green leaf significantly more closely than did to the soil background. In terms of whether grasshoppers tend to land on a leaf background after they're disturbed and engaged in the display, we found that grasshoppers were more likely to land on matching background after the display behavior, again using the GLMMM. And then in terms of flight initiation in relation to background matching, we predicted that the grasshoppers on unmatched soil background will initiate the display at a greater distance without allowing contact, and we found that to be the case. There was significant difference there. And in terms of whether the grasshopper would tolerate the observer actually coming into contact, coming close enough to actually contact the grasshopper before initiating the display, that was frequently the case when they were on a matched background, a green leaf background, but never the case when they were on the unmatched background or soil background. So again, that prediction was borne out. And then if looking at the prediction that deimatic displays are energetically costly, so that grasshoppers will reduce their travel distances. Sequential displays, we found that to be the case as well. And so just to wind up here, Paramastax nigra, this South American grasshopper, shows clear evidence of being camouflaged against a green leaf background with respect to luminance. Individuals adjust their probability of initiating display depending on their match to their current background. They initiate the display at a longer distance when they're on an unmatched background, and they never allow approaching observers to make contact before initiating a display from an unmatched background. Individuals decrease the length of their escape trajectory across multiple displays, demonstrating a likely energetic cost of behavior. So, at least from these preliminary results, we infer that the escape behavior of Paramastax nigra is likely to be a deimatic display. And so I'm going to stop there and open it up to questions. Without a cattail.
Yes, she was taking a nap and now she's off.
Okay, still got a little time for questions, but if Muhammed Shariq, if you want to put up your first slide just to get ready. We'll wait until the designated time to start because we want to keep, there'll be people coming in from other sessions, and we want to make sure that the timing lines up. Okay, our next speaker is Muhammad Atif Shariq. We'll be talking about detecting adaptive genetic variation in light of ecological interactions.
Thank you, Kyle. Thank you to the organizers for this opportunity. So today I'll be talking about some proof of concept ideas, some methods in trying to detect adaptive genetic variation in certain ecological interaction scenarios. To begin, hmm, yeah, so trait variation is essential for populations to adapt to changing environments, and the heritable component of trait variation, otherwise the genetic component, is a key part in supporting species survival and adaptation, and thus biodiversity. Without this, species are at a risk to run into extinction. However, whether heritable trait variations drive adaptation depends on whether the variation is neutral or adaptive. So, to briefly tell you the difference between the two, neutral variation is caused by genetic drift and mutations, and they usually have little to no effect on fitness, um, except in cases where uh, through mutation events and recombination, there is certain variation, and thus it can affect fitness of the population. And adaptive variation is usually driven by disruptive selection, and it directly has a great effect on fitness. Um, Comparing between, telling them apart is a key question for ecologists and evolutionary biologists. And one of the method to tell them apart is by comparing QST, or the quantitative trait differentiation, to the fixation index, to the FST. Going ahead, so what is this QST and FST? To understand it better, I have a simple graphic where we can see what QST is. So QST is basically the measure for quantitative trait differentiation, and this is the formula for it. It's basically the genetic variance between patches to the total genetic variance that we see in the population, which is basically the additive genetic variance between patches and uh, the genetic variance within habitat patches. While QST is the measure for quantitative trait differentiation, FST measures the differentiation at neutral genetic loci. Now, to compare QST and FST, we basically interpret a similar QST and FST value in the population as a sign of neutral evolution. And thus, Whitlock in 2008, when plotted neutral loci marker, that is FST, and QST, or the neutral traits, has seen that a very similar plots for in both these cases. And thus, this has been shown to act as a good statistical null in such methods. However, a QST greater than FST is a classic sign for spatially disruptive selection. To understand it better, say, for example, we have two different seed sizes in two different habitat patches, and thus birds of that habitat patch will more likely to be adapted to that seed type. So, phenotypically, um, could be genetically. And this basically is seen by increased between patch variance, and thus a high QST value, and thus giving us the QST greater than FST signal, pushing the peak towards the right. A QST, oh, I'm sorry about this. This shouldn't have happened. Well, let me just talk about this. So a QST lesser than FST signal is basically a sign for stabilizing selection. So here we basically see that, say, for example, there is the same seed size in all the habitat patches, and so birds of different habitat patches will be adapted towards having just one seed size, for example. And through decreasing this between patch genetic variance, QST is also going down, and thus we get a QST lesser than FST signal in this scenario, and it will basically push the graph towards push the peak towards the left. However, is this all that we know? It is all that there is to it. We suggest that there might be a couple of more ecological scenarios where QST lesser than FST it can be seen by increasing the within patch genetic variants. To name them, it's basically locally disruptive selection and temporally disruptive selection. Locally disruptive selection is also called as balancing selection or negative frequency disruptive selection. In a scenario where there is a distribution of resources in the habitat patch, where there are, say, different sizes of the seed, or in a temporally disruptive selection case where there is a temporal variation in resource type or resource property, where you have, say, for example, interannual fluctuations in seed sizes, within the habitat patch, we see that there is some sort of a variation. We expect to see that there is some sort of variation in individuals in that habitat patch. And they are basically trying to exploit one part of resources, and thus we see that many different phenotypes are equally present in the population. And this gives us a QST, and this we hypothesize that we get a QST lesser than FST value because there is a increased within patch genetic variance in these scenarios. And we have also seen this grant, and this is by increasing within patch genetic variance and thus decreasing QST value. And we have seen this in Darwin's finches as well, where on the same island there are two different phenotype of birds, one adapted to having small seeds and the other phenotype having adapted to eat the larger seeds, and they are of the same species. Now, to test this, we are using individual-based simulations. So it's completely in silico, and we use a software called Nemoage. Refer to the paper to know more about the software. It's really cool. And we have certain we have a structured population, and they have undergo these life cycle events, and we simulate it for 20,000 years to reach a balance. And basically what we're trying to do is we're trying to simulate different genetic and different diversifying forces on separate selection regimes, as we have spoken before, and then we calculate the QST and FST from the results that we obtain from these simulations. And for the statistical analysis, we use the classic Levant and Krakauer approach. It just works for our case. Going to the it's very important to note that it is just a proof of concept idea for introducing different selection regimes to interpret a QST lesser than FST result. So coming to the results section, we see that on the y-axis, I have plotted the statistical power basically out of 100 replicates, in how many replicates we get a QST-FST comparison as per our hypothesis. We see that in the case of neutrality, we get a quite strong QST equal to FST result. In case of spatially disrupted selection, we get a QST higher than FST result in almost all the cases. However, in stabilizing selection, we get quite a weak QST lesser than FST signal. This is quite apart from what the general idea of QST lesser than FST signal is. However, previously done simulation studies have also found it difficult to obtain a QST lesser than FST signal by solely applying stabilizing selection to the population. However, in both the cases of locally disruptive selection and temporally disruptive selection, we get quite a strong QST lesser than FST value, and thus confirming our hypothesis. However, it is important to note that this is at a low migration rate where only 1% of the population within the habitat patch is migrating to the other patches. When we have also tested for higher levels of migration, say for 10% and 50% of the individuals migrating between habitat patches. And in these scenarios, we obtain a weaker signal in almost all cases, as expected, because basically migration is reducing the strength of the diversifying selection pressures in the populations. But it's important to note that at a high migration rate, say 50% or more, and also at when the sampling size is small, which is where there are very few individuals being tested, we get a somewhat strong QST lesser than FST signal, even in the case of stabilizing selection. And to conclude, we see that a QST lesser than FST value is difficult to obtain through just stabilizing selection alone, except for the scenario that I mentioned earlier. However, in the case of locally disruptive selection, or say negative frequency dependent selection, uh, we have confirmed and we get quite strong QST lesser than FST signals. And same goes with temporally disruptive selection, where there is temporal variation in resources within patches, and thus having these forces on acting on the individuals. So basically, I want to leave you with a take-home message saying that we need to consider alternate interpretations and look more closely at QST lesser than FST signals in our empirical studies as well. And with that, I'd like to conclude the talk. I would like to thank the funding agency, my home university for housing me, and also all the members in the lab. Thank you very much. And I'd like to take some questions if you have any.
Okay, thanks very much. So have a bit of time for questions. Okay, Sam, if you want to unmute yourself and go ahead and ask your question.
Yeah, so how does this compare with uh, methods for directly measuring selection rather than just looking at trait variances?
I'm sorry, I don't think I understand the question.
So,
Uh,
so you're talking about estimating selection by measuring variances, QST, FST, but then you can also just directly measure selection, right? Using like Landy-Arnold approaches. So how do these measures compare with those? Are they complementary? Could they help sort out some of these ambiguities?
um, so the thing is, well, the previously stated results suggest only that there is no diversifying force at play, or say there is no disruptive selection in the population when we see a QST lesser than FST values. And yes, even though we can measure selection directly in the patches, we rarely go a step ahead and also interpret um, additive genetic variants at play in these patches or in the population to connect it to uh, the selection in the thing. So in the system. Um, and so it is. So what we are trying to do is basically simulating through the other way around, where we interpret where we introduce the selection pressures and then look at the additive genetic variance patterns in the population.
Okay, we're gonna have to
Uh,
leave it there because we
yeah,
have to move on. If you want to continue the discussion
all
in the
right.
chat,
I
but
can chat with you.
okay,
Thank you very much.
thanks very much. So our next speaker is Deidra Jacobsen. Talking about shifts in stem structure and floral allocation under abiotic and biotic stress. Deidra, are you there?
Yes, when I shared my screen, Zoom closed out. So let me try that again.
Okay. Okay, we can see you. Awesome. Okay, great.
Okay. Are you able to see me?
Yep.
Okay, perfect. Make sure. All right. Am I good to start? All right, thank you. So
Good to go.
today I'm going to be talking about some shifts in stem structure and floral allocation under abiotic and biotic stress. This is work that was done by a couple undergrads in my lab, Lily Ruckhouse and Henley Kraus. So all organisms, including plants, have limited resources to allocate among growth, reproduction, and defense. And how plants allocate those resources affects survival and fitness and is also dependent on a variety of internal and external factors. So under stressful conditions, plants often have to prioritize survival by shifting resources toward defense and away from growth and reproduction. The overall effect then is that stress limits overall resources and shifts resource allocation among categories. So in stressful growth conditions, plants are often smaller with lower fecundity and reproductive output because they have to prioritize defense and survival. Plants experience simultaneous abiotic and biotic stressors, and different types of stress trigger specific plant responses. Under drought, a plant's goal is going to be to decrease water loss and try to maintain trigger pressure and avoid cavitation. So some of this might be accomplished by closing stomata and investing in roots. Under herbivore attack, the goal is to stop further attack. This could be by increasing defensive compounds to minimize tissue loss, and this can be by attracting predators to keep herbivores in check, and by also shifting resources below ground to keep some of those above ground resources safe from above ground attack. Coordinating these different stress responses can therefore lead to conflicting pressures on resource allocation. And we know that a lot of this response is hormonally regulated by signaling pathways involved in both growth and defense. So abscisic acid is involved mostly in drought regulation and osmotic acid in insect stress, but we know that there's a lot of crosstalk among these pathways. Less is known, however, about how combined stresses reshape whole plant allocation, architecture, and reproductive investment. So one problem with answering this question is that we often like to do experiments that quantify fecundity without capturing structural plasticity. So for example, in many experiments, we count flower number, seed set, plant size without capturing other plant traits that affect survival and interactions with mutualists and antagonists. However, if plants are changing in structural traits like height, that not only affects growth investment, but also potentially alters visibility and appearancy to insects and pollinators and can hint at underlying physiological changes. In particular, stem diameter is a really incomplete picture of plant size and growth. Although we often use stem diameter as a proxy for plant size, if we look a little bit deeper, internal stem anatomy reflects not only growth investment, but can also reflect internal changes in water and nutrient transfer capabilities and a plant's ability to withstand mechanical stress. So for this experiment, we tested the prediction that both drought and herbivory will reduce plant size and floral allocation, but have different effects on plant structural traits. So for this study, we did a greenhouse study with a 2x2 factorial manipulation of drought and herbivory to create 4 groups of plants. So one was a control, one was a drought only that received less water, herbivory only treatment. This was a set of plants that received induction with a spray of jasmonic acid to simulate herbivory. So this was giving them sort of the first hormone in this cascade of defenses that plants produce following herbivory. So they think they're being herbivore damaged without us having to actually introduce herbivores into the system. And then the final treatment was a combination of both drought and insect herbivory with jasmonic acid. And the system that we used for the study was Oenothera harringtonii, or evening primrose. And the species is a short-lived Colorado endemic that's pollinated by hawk moths like Manduca and Hyles lineata. Hyles is really interesting because the adults are pollinators, but the larvae also act as herbivores on these same plants. And then the reproductive measurements for this study were taken after we had applied the treatments and let the plants grow for a bit. This was done by my undergraduate, Lily. Some of these measurements included flowering time, flower size, and flower number. We also took some male allocation measurements like pollen viability, but those results are still pending. Another student looked at above and below ground allocation. We measured stem length, stem diameter, and root biomass. So in addition to using stem diameter as a proxy for plant size, we also looked at stem tissue layers by harvesting plants at the end of the experiment. So a quick reminder of Plant Physiology 101. This includes xylem for water movement and phloem for nutrient transport, as well as lignified tissues that are involved in support. So once we harvested the stems, we did a staining of cross sections with help from a colleague, Dr. Dan Gladish, to identify some crude stem sections associated with different physiological and structural traits. So we used fluoroglucinol stain. This stains xylem and secondary growth red, but phloem doesn't stain, so we can help sort out those layers. We identified a few main sections to measure. This included an outer layer of secondary phloem and cortex involved in nutrient transport and structure, and then an inner ring of woody secondary xylem that contributes mostly to support and water transfer, and then the central pith layer. I'll walk you through the individual results on the following slides, but the overall result here was that abiotic and biotic stress had different effects on reproduction. Drought reduced mostly overall floral allocation, while herbivory mainly affected flowering time. So starting first with the effects of drought, drought reduced both flower number and flower size. Here I'm showing you on this graph flower number with more flowers in the well-watered treatments and reduced flower production under drought in the yellow and orange treatments. We see the same pattern for flower size with longer flowers in the well-watered treatments and shorter flowers in the yellow and orange drought treatments. For herbivory stress, the main result was that induced plants took longer to flower, and this is seen here as an increase in the number of days to flowering in the red and orange induction treatments. So now shifting to look at plant growth and size, we also found that stress altered plant size and structure. As I'll show on the following slides, the overall result here was that there were no interactions between stressors. Drought mostly affected growth, while induction changed resource allocation. The first result was that drought altered external stem traits, such that drought-stressed plants were shorter and they were wider. On this graph, I'm showing stem compactness, meaning how tall and skinny they were versus short and wide. So on the graph, drought treatments in yellow and orange are shorter and wider than the well-watered plants. And this is consistent with an expectation of compact architecture under stress. This might help plants structurally, as well as create a sort of microhabitat that conserves humidity around more tightly clustered leaves to help minimize some of that water loss. And at the end of the experiment, there was a convergence of stem diameters across treatments, but we did find some differences in internal stem traits when we harvested plants for processing. Looking at internal stem structure, we found that drought altered the thickness such that the outer stem layers were reduced. So the thinner outer phloem and cortex layers were in the yellow and orange drought treatments. This is probably consistent with lower cell trigger pressure and reduced cell expansion under drought. This also likely reflects reduced nutrient transport due to a thinner phloem layer. In contrast, induction didn't really alter stems as much. The only real change was a trend that induction increased the thickness of inner layers. So this is shown on the graph as a small increase in the thickness of the inner woody secondary xylem layers in the induction treatments in red and orange. This could be consistent with some mechanical and structural reinforcement under insect attack. The main effect of induction, though, was increased below ground allocation. So overall root biomass was slightly higher in induced plants. This is shown on the graph as more root biomass in the red and orange induced treatments. Because Oenothera grows as either an annual or a short-lived perennial, it's possible that when they're under above ground attack, plants shift resources below ground to try to hide out and try again in future years. However, we do see a lot of variation across systems and how plants distribute above and below ground resources under stress. This is likely going to be dependent on the level of drought and herbivore attack. For this study, though, the main take-home is that drought reduced plant size and flower production, producing more compact plants with reduced outer stem layers, while induction maintains size but delays flowering and increases below ground investment. So although we expected an interaction of these stressors, plants really responded in different ways and independently to the abiotic and biotic stresses. So this work's relevant for understanding how plants adapt to drought, which is becoming more frequent and severe under climate change. And it also highlights how abiotic and biotic stresses can both reshape growth and reproductive allocation, and not just in the short term, but in ways that may influence broader stress responses, especially for plants that might have variation in the number of years that they're surviving and reproducing. These plastics changes also connect to the evolution of life history trade-offs, where plants are balancing investment in survival versus reproduction under variable and often simultaneous stresses. And then together, this links some of these immediate stress responses to those longer-term ecological and evolutionary outcomes. And so this work, like I said, was done by a couple of students in my lab. We were also assisted by Dan Gladish, who helped with some of the sense staining and project planning. We have a great group of students who help take care of the plants at the conservatory, and Robert Gusso and Chris Escogan donated some seeds for this project that was funded by Miami University. So I think I have some time at this point to take questions if anyone has any.
Yes, thanks. If there are any questions, please go ahead.
I had a question. So did you say that these plants can be either perennial or annual?
Yeah, it depends on probably a lot of things like drought and herbivory.
Right.
So there is some variation. I think it also depends on temperature and climate. They don't have a broad range, so there's probably not a lot of range variation, but like year to year variation.
Okay, yeah, I was just wondering if you knew like which your populations were. Like, do you know if those were perennials or
That's
annual?
a good question. We had plants from a couple different populations. I'd have to look back at where they came
Yeah,
from and see if there was variation. We didn't have a ton of plants overall, so if there was, we might not be able to detect it, but that's a good
right.
question.
Okay, yeah, I was just curious about like whether that would affect like the direction of the responses, but
Yeah, I
yeah,
think it would for
cool,
sure.
cool talk.
Okay, Arash, if you want to go ahead and share your screen. And yeah, we'll start up in just a minute.
Okay.
Okay, our next speaker is Arash Raeisbahrami, and he will be talking about a quantitative framework for local adaptation, revealing pervasive trade-offs and widespread asymmetry.
Okay, thank you so much. For this presentation, I want to talk about local adaptation and its underlying mechanisms. But first, what is local adaptation? So imagine we have two populations of flies living in cold northern part of the US and warm southern areas. We can test for local adaptation with reciprocal transplant experiments, where we put the flies from warm environments in the cold temperature and put the flies from the cold environment into the warm temperature and then compare their fitness. So local adaptation happens when a population shows a higher fitness in its native environment than the other non-native population in the same environment. So here we are observing local adaptation for both populations because they have higher fitness in their native environment than the other population in the same environment. So local adaptation is a very important process in evolutionary biology because it ties up two important concepts of divergence and adaptations, because it maintains the divergence of populations under gene flow through adaptation. Okay, but what causes local adaptation? So the most well-known mechanism of local adaptation is fitness trade-offs. So what is fitness trade-offs? If a population is so adapted to its local environment that its traits are disadvantaged in the other environment, we can say that this population is showing fitness trade-off. So here we are seeing a fitness trade-off for again both populations, which have significantly higher fitness in their native environment than in their non-native environments. But how exactly fitness trade-offs causes local adaptation? So for example, here the local adaptation of population 1, which is an aphid inhabiting alpha-alpha, is because of the fitness trade-off of the other population, the clover aphid. So, and this is the same pattern of fitness trade-off can be observed in the other population. And this is how we mainly think about local adaptation and the effect of fitness trade-offs. And this is the assumption of most classic methods. But fitness trade-offs is not the only mechanism of local adaptation, because not all cases of local adaptation are this symmetric. What do I mean by this symmetric? So there are two main patterns of local adaptation. The first pattern is symmetric local adaptation. Continuing with our aphid example, both host races are showing a similar magnitude of local adaptation. So local adaptation here is symmetric. The second pattern is asymmetric local adaptation. In this case, one population, so for example here, a snake with grasshoppers living on goldenrods, shows a significantly higher local adaptation than the other host race that lives on snake with and exhibits no local adaptation. So, as we can observe, the main driver of symmetric local adaptation is fitness trade-offs. But the problem arises when we consider asymmetric local adaptation. In the asymmetric cases of local adaptation, we usually do not see that fitness trade-offs underlie local adaptation. This means that in the asymmetric cases, another mechanism should be involved. This mechanism is general fitness advantage. So, for example, here is the result of another asymmetric local adaptation in Prunella grandiflora and Prunella vulgaris. Here, the local adaptation of Prunella vulgaris cannot be due to fitness trade-off of the grandiflora, because the degree of fitness trade-off is very small in comparison to the large magnitude of local adaptation. So what produces local adaptation here? We see that Prunella vulgaris is performing better than grandiflora even in its non-native environment. So vulgaris has a general fitness advantage that causes this local adaptation. How we can measure this? By comparing the fitness of both populations in their native environments. Why does this difference show the general fitness advantage? Because of an assumption that we have. We are assuming that populations in each environment have reached their optimal phenotype. So the fitness of a population in its native environment is the highest fitness possible of that population. So with this assumption, comparing the fitness of populations in their native environment show the general fitness advantage or lack of it. So now we have two mechanisms that contribute to local adaptation. So here I first introduce our proposed framework to measure the relative contribution of each mechanism to local adaptation, and then apply that framework to publish the studies of local adaptation. So imagine the degree of local adaptation in population 1. It's equals to the fitness of population 1 in environment 1 minus the fitness of population 2 in the same environment. If we just add the value of the fitness of population 2 in the environment 2 and then subtract the same value, nothing mathematically should change. But then we can rewrite the same equation like this. So now this equation has two biologically meaningful components. One for general fitness advantage and the other one for fitness trade-off. What does that mean? It means that we just show that the magnitude of the local adaptation of one population is mathematically the sum of general fitness advantage of that population and the fitness trade-off of the other population. So now the easiest thing is to just calculate the contribution of fitness trade-offs to local adaptation just by dividing the degree of fitness trade-offs of the second population to the overall magnitude of the local adaptation of the first population. This ratio can technically range from negative infinity to positive infinity, but several values are critical. So the value 0 is an important case where there is no fitness trade-offs and the only underlying mechanism is general fitness advantage. Any value between 0 and 1 shows the relative contribution of fitness trade-off to local adaptation. Value 1 shows cases where local adaptation is only due to fitness trade-offs and general fitness advantage does not exist. And any value higher than 1 means that the magnitude of fitness trade-off is larger than the magnitude of overall local adaptation. This means that either our focal population has a general fitness disadvantage or the environment is so harsh that both populations are performing poorly, so the magnitude of overall local adaptation is reduced. So now we have a framework to quantify the magnitude of local trade-off, patterns of local trade-off, and the contribution of different mechanisms to local adaptation. Local adaptation, yes. With this framework, now we can ask new questions and reassess the patterns of local adaptation we observe in nature and lab experiments. For this, I conducted a meta-analysis on 498 published reciprocal transplant experiments and extracted the fitness of populations in their native and non-native environments, transferred the fitness values into relative fitness values, and then compared them. I used this data to ask several questions. The first one is, how prevalent is local adaptation in general? The results show that almost 70% of all the cases show signs of local adaptation, and the overall average of magnitude of local adaptation is 0.18. This shows that, consistent with the previous studies, local adaptation is a common phenomenon. Our second question is, are the cases of local adaptation symmetric or asymmetric? We are seeing that that's among 354 reciprocal transplant experiments that show signs of local adaptation, at least for one population, 65% of them show asymmetric local adaptation. And the average degree of asymmetry is 0.41, which is almost high. These results show that the asymmetric local adaptation is very common, which is a sign that having a quantitative framework to measure relative contribution of different mechanisms to local adaptation is actually very important, because most of the cases are actually asymmetry, and we need a good framework to account for that. So now we can just explore the relative contribution of fitness trade-offs to local adaptation. The results show that from 478 populations that show local adaptation, almost 77% of cases show signs of fitness trade-offs involved. 58% show signs of general fitness advantage, and 35% show a joint contribution of fitness trade-offs and general fitness advantage. So we can just conclude that fitness trade-off is a major factor driving local adaptation. But what about the mechanisms underlying symmetric and asymmetric local adaptation? Before we dive into the results, I just want to briefly note how I handled the math here, because the contribution of fitness trade-off is calculated as a ratio. It creates a heavy tail distribution. To account for that, I use the Bayesian framework to first estimate the joint distribution of underlying fitness components, and then drive the final distribution of the contribution of fitness trade-offs to local adaptation. But what's the result? The results show that the contribution of fitness trade-offs to local adaptation is significantly lower in asymmetric local adaptation, which is consistent with our previous expectations. Also, we are seeing that as the contribution of fitness trade-off increases, the degree of asymmetry decreases, and local adaptation becomes more symmetric. These results show how the mechanisms underlying local adaptation act differently among different patterns of asymmetry or symmetry. And finally, I want to ask how local adaptation and its mechanisms behave across a speciation continuum. To do this, I categorized each pair of populations into early, medium, and late stages of divergence, and investigated the degree of local adaptation in each category. I specified these categories based on the description of the population pairs in the original publication. The results show that the magnitude of local adaptation is significantly lower in the earlier stages of divergence than the medium and the late stages. The contribution of fitness trade-offs to local adaptation across these stages is constant and high, around 1. This shows that fitness trade-off is the main cause of local adaptation across all stages of divergence, but these results also show that this value is maximized in the medium stages of divergence. But the symmetry-asymmetry pattern of local adaptation do not differ significantly among these stages, although we are seeing a continuous increasing trend, but this is not statistically significant. In the end, we are proposing a framework that can tease apart and measure the relative contribution of different mechanisms involved in local adaptation. We found that the majority of populations are showing signs of local adaptation. The majority of local adaptation cases are asymmetric. Fitness trade-offs is the main mechanisms involved in local adaptation, especially in symmetric cases of local adaptation. And local adaptation is more pronounced in later stages of divergence, while fitness trade-offs are more important in the medium stages of divergence. And the pattern is not that different. With that, I want to thank George Washington University, Lini Zhang's lab, and take any questions. I can't hear. Sorry.
Thanks very much. We've got just a little time, so only maybe I have one quick question, but yeah, Payal, if you want to go ahead and share your slides, and we'll get started with that talk in just a minute. Okay, Scott Winters, did you want to unmute and ask a quick question?
I think their question is in the Q&A. I can read what it says. What are the implications of your work to the understanding of outbreeding depression? particularly in light of conservation/restoration interventions such as genetic rescue or assisted gene flow? And
It's a good question.
it's
It's
2:30, so if you want to
yeah,
take your answer. Yeah,
yes,
thank you.
okay. Yeah.
Perfect.
Yeah, go ahead and type your answer in the chat, and we've got to move on to the next talk from Payal Dash. We'll be talking about environmental DNA. Payal, if you want to go ahead, your talk starts now.
Hello everyone, I'm Payal Dash. I'm a PhD student in Jagiellonian University in Krakow, Poland. And today's topic of my discussion is environmental DNA illuminates patterns in targeted communities, evolution patterns in targeted communities across urban and elevational gradients. Before that, let me recap what we know about tardigrades. They have an ancient evolutionary history and they are known for their cryptobiotic abilities. They can withstand extreme conditions, yet they have a potential as bioindicators. They are found in a variety of environments and they are quite diverse and have the ability to disperse. Thus, they serve as key models for micrometazoan biodiversity and evolution. For the longest time, tardigrade species were predicted to be widespread, following the everything is everywhere hypothesis. But in reality, most tardigrade species have restricted distributions. We know less about what happens at the regional and local levels, but we know that these patterns exist for the tiny animals, but at a micro scale, which quite similar to larger animals. So the broader question is, how does environment affect tardigrade communities regionally and locally? We study limnoterrestrial tardigrade communities, the communities which are found in moss and lichen ecosystems. And we plan to do the study at a local level as well as a regional level. For the local level study, which I'm going to be focusing more on today, we collect tardigrades communities from different sites and look at habitat level variations in microclimate, elevation, and urbanization. To ask how environmental variations affect local distribution of taxa. For the regional level study, we focus on our focus is on the Gibraltar Strait region, and there has been a lot of changes in sea level and appearance of mountains. So this gave us the rationale of asking like how historical changes affect the current distribution across the Gibraltar. Coming to the local level study, we submitted our manuscript last year, end of the last year, which was accepted this month, where we investigate tardigrade communities across elevation and urbanization zones with environmental DNA. For this, we had collected samples from southern Poland in the month of April during spring. We collected combination of moss and lichen samples along with environmental data. We used more than one method to study these communities. We used the traditional microscopy-based identification methods, where we identified tardigrades taxa based on cuticle sculpturing, pharyngeal placoids, buccal apparatus, claws, and other features. And in addition, we also used advanced methods, which is the environmental DNA or the eDNA methods, which is the DNA obtained directly from the environmental sample. It is much faster, requires less expertise, and is high throughput. So it provides more clear advantages over the traditional methods, but we use both in our study. So we use these methods to test the following hypothesis. The first one is that the eDNA-based method captures a greater diversity of taxa compared to traditional method of identification. And second, that elevation and urbanization affect different tardigrade taxa in different ways, causing changes in community composition across sites. Before starting, we tested different eDNA extraction methods and found that the home brew method, which is the mu-DNA method, was performed better than the commercial kit. And it is an advantage as it is much cheaper to collect eDNA from using this method. Using this mu-DNA method, we collected eDNA and also sequenced amplified the CO1 and 18S markers, and using the CO1 and 18S markers. Alongside, we also did the morphology-based identification. Simultaneously, we also used this reference barcode to which would help us identify the taxa. The sequencing was done on an Illumina MESEC platform, and following the when we obtained the sequences, we followed a metabarcoding pipeline for communities, where we followed the steps of quality filtering, assembly, motive delimitation, and taxonomic assignments, giving rise to the eDNA-based datasets. So in total, we had 3 datasets. One is the morphology-based identified taxa, and the molecular OTOs obtained from the eDNA datasets. Using these, we performed the community environment correlation analysis, where we calculated richness, diversity, number of unique genera across methods, and performed different tests to understand the community environmental correlation. So these are the key results from our study. We found that each method recovered some unique genera, but the highest number of unique genera were recovered by the 18S eDNA dataset. And this was a summary statistics which said the same. When we looked at the prevalence weighted abundance across methods, we found that each method recovered some unique genera, and out of all the methods, 18S recovered the highest number of unique genera across both elevation and urbanization zones. This also proved our first hypothesis that eDNA is actually working better than the morphology-based identification. When we looked at genus richness across methods, we found that forest localities recovered had a greater genus richness than city localities. It gave rise to the results that there is a moderate influence of urbanization on tardigrade community structure. And we also found that out of the rare groups which occur only in occurring in just one sample, out of 10, 7 were found in low elevation forest zones, indicating a ecologically important zone. These results show the influence of a habitat type on community composition. When we looked at the number of unique genera across methods, we saw that the differences were more pronounced between city and forest sites than between high and low elevation zones. From the environmental correlations, using distance-based redundancy analysis, we found that there were significant correlations with microclimate, with temperature and humidity especially, in different combinations of data sets. Humidity was the dominant environmental driver in molecular assessment, and temperature was key environmental gradient in the morphology-based assessment. And we also found that turnover explained more variation than nestedness. When we looked at the explained variance by the environmental variables, we found that the explained variables by the zones and the microclimate were significant in different combinations of data sets again. And it supported our second hypothesis that elevation and urbanization do affect tardigrade taxa in different ways, causing changes in community composition across sites. So the methodological and biodiversity insights we obtained from our study was multi-marker eDNA and morphology have give rise to complementary strengths, and together they gave a very complete community picture. Our home brew method was proved to be better, and 18S was more reliable than CO1 when it comes to the performance. 18S marker gave the highest motive recovery, and it detected rare taxa, and it had the strongest correlations with humidity and temperatures. CO1 explained the highest variance in the distance-based redundancy analysis, and it gave rise to a finer taxonomic resolution and was very consistent across replicates. Morphology was also reliable. It detected tardigrades in more samples, and it shows a clear advantage in some ways. But this is limited by cryptic speciation, cryptic species, and also outdated descriptions. Genetically distinct motifs revealed cryptic diversity. And in the end, we concluded that eDNA analysis is feasible, but it requires more updated references databases for accurate identification. So in all, we found these methods revealed hidden diversity and community patterns. Humidity and temperature were the strongest predictors, and this may be true because they are important factors that regulate activity, reproduction, survival of tardigrades. Elevation showed method-specific patterns, but it is clearer in morphology. Urbanization reduced richness, but causes homogenization. It could be attributed to pollution-driven loss of rare taxa. We found that turnover dominates nestedness, and the sites, it indicated that populations may be undergoing local adaptations to microclimate, climate niches. In the end, we conclude that integrated approaches are needed, required to resolve environmental filtering and community assembly, and more than one methods must be used to have better conclusions. So this study gave a robust framework for understanding tardigrade diversity patterns across zones and gradients. The ongoing work and future directions are incorporating long-term temperature data and also doing a seasonal study, which we are doing currently. And we are also planning to do the regional based study in the future, where we plan to do the phylogeographic analysis and also identifying putative novel taxa especially from the Morocco region, because it is underexplored. So I want to thank you for your attention and thank the organizers for giving me the opportunity for this talk. Thank you.
Okay, we've got a little bit of time for questions. So if anyone has questions, either unmute and ask away, or you can type in the Q&A. Elizabeth, if you want to unmute and ask your question.
Yes, Payal, great talk. And I had a question. Are you one of the first people to do eDNA of tardigrades?
I mean, just eDNA, yes, I think it is not something which has been done, and a lot before, and yeah, using individual tardigrades obtained from environmental samples, sequencing has been done, but from the environmental DNA, this is a method which we have developed, and we have done it for the first time. And this is a problem because eDNA based studies are difficult, as the references databases are not updated, and this is our also a goal to generate more sequences and update the reference databases so that such kind of studies can be done better in future.
Well
All right, we're
done.
going to have to leave it there and move on to our next speaker. Liz, if you want to put up your screen. And so our next speaker is Elizabeth Mandeville, talking about hybridization and leuciscid fish hybrids. And take it away.
Hi, yeah, I'm Liz Mandeville, and I'm going to be talking about some work that my lab has been doing about the phenotypes of hybrid fish. And this is work that started at the University of Guelph in Ontario and has continued on to my current position at Northern Michigan University. So it's been a really lovely integration of effort by a lot of these lovely people. These are all current or former students in my lab. Oh, actually, no, all of them have graduated. Happy for them, sad for me. So this work was initiated by graduate students Amanda Muser and Amy Pachira, and has been continued by undergraduates, Eric and Amanda. Okay, so the first piece of background you need for this is that hybridization is super common in nature, especially in freshwater fish species. And so this picture shows one reason for that. Spawning looks like this. So a lot of fish species have external fertilization. Some species don't build their own nests. And so you have multiple species spawning together at the same time in the same location, which tends to lead to hybrids for closely related species. This has been known for quite a while in various fish clades. The other lovely thing about fish is that there are about 30,000 extant species. So there are a lot of clades of species that have a whole bunch of species represented in the same geographic location. I'm going to focus today on the minnows formerly known as the Cyprinidae, now known as the leuciscidae. These are things like common shiner, creek jub, western black-nosed dace. There have been studies that there are hybrids between these fish for some time. The figure I'm showing now and the cool photos of spawning fish are from a paper by Kurush et al. In 2021. And this paper is interesting because it suggests that hybridization might be driven by breeding behavior. But it also inspired us to look more closely at hybridization from a genomic perspective in these regions. A crucial thing to know at this point is that prior to our work, most of the work that had been done on hybrids in these clades was based on phenotypic analysis of the fish, or traits, where hybridization was inferred by phenotypes. So we further hypothesized that there might be a gradient of hybridization outcomes along gradients of anthropogenic disturbance. So like many portions of the world, including the Midwest and the United States, Southern Ontario has experienced a lot of agriculture and urbanization. And so to me, this suggested the potential for hybridization. The mechanism for increased hybridization after disturbance would be either increased opportunity for hybridization, either due to a narrower time spawning window or fewer suitable sites, or perhaps the fitness consequences of phenotypes have changed and the hybrids have some phenotype that makes them have higher fitness. Could be either, we don't know. And so we set out to characterize hybridization in these fish. We originally targeted 9 species in 29 locations. These students worked really hard, got a great sample size, and these locations span a range of anthropogenic disturbance. We then generated genotyping by sequencing data. For those who are keen to know more about this, we have a recently published paper in Molecular Ecology with Amanda Muser as the first author. I'm going to go kind of fast through this so I can get to the phenotypes, which is the main point of my talk today. So we sampled a whole bunch of different locations with a range of disturbance. We kind of thought that we would see a signal of disturbance. Spoiler, we didn't really. That's part of the story that you'll get from Amanda's paper. But I'll get to what we did find. Okay, so also for those who work on these species, we did generate a novel reference genome as part of this work. This is a resource that's now available for anybody who wants to work on these species from a genomic perspective. Okay, so we went and sampled all these fish. We estimated ancestry of each individual fish using a hierarchical clustering algorithm called entropy. This is similar to things like structure and admixture. We're using this reduced representation genotyping by sequencing data to infer what percent of each individual's ancestry comes from each species. And so from this, we found that 29% of the individuals we had sampled were hybrids. That's high. That's really high. And so we found also that there were different suites of species at different sites. This is a small multiples plot where we have each site and each species is a color. Again, this is in the paper if you want to squint at it more closely at length. But the quick story is that there are two species hybrids, a lot of them, and a few multi-species hybrids. It also wasn't restricted to first generation hybrids. There are a lot of back crosses or advanced generation hybrids as well. And so it looks like hybridization in this group of fish is pretty widespread, pretty rampant. I'd love to be able to tell you why, but that's not the story I'm telling today. That's the story I hope to be able to tell in a few years. And so something funny about this, though, is that we didn't call any of them hybrids in the field. Now, this is not an easy group of fish. They all look kind of like this. They're small, they have a black stripe down their side. There are characteristics to tell them apart, but for rookies, it would be reasonable to make mistakes. However, the people identifying these were well trained and excellent at it, Amy Pachura in particular. And so we looked back through the photos and we all thought, no, these still look like parental species. But the fact remains that we had all these hybrids and we were pretty confident in our evidence for hybridization too. And so just to summarize a little bit further, every species we sampled hybridized with at least one other species in our sample. Some of the crosses were more abundant than others, but we had this incredible fluidity of species boundaries in this group. And so that's where I'm going to stop talking about genomics for today, but if you're interested, we wrote a paper on it. It's out in the world now as of about a month or two ago. But what we started wondering is, how did we miss all these hybrids? So, we looked back at the photos and we were still pretty confident in our phenotypic IDs, but we did also trust our genetic IDs. There's been a really wonderful literature developing the last few years about the phenotypes of hybrid individuals. As we've gotten more genomic data, we've been able to identify more hybrids. And so there's this circular factor where hybrids were previously identified morphologically. As we can sequence more of them, we identify more hybrids. And so we're completing that circle and going back to our genomically identified fish and characterizing their morphology to try and understand, what we missed. What are the phenotypes of hybrids? Are they like a parental species? Are they intermediate? There's really good meta-analysis a couple years back suggesting that actually hybrids are more often closer to one of the parental species, but obviously that varies by clade. And so we characterized a number of key features that are the sorts of traits that you would use in the field to identify these fish. So we could also take a geometric morphometric approach or something like that, but we started with the things that you would do if you had the fish in hand in the field and were trying to identify it. So we characterized the mouth morphology. We characterized fin ray counts. We characterized lateral line scale counts, or the number of scales on the side of the fish along this line of pores called the lateral line. So we started to have the results roll in this spring, and it's pretty cool. This is work in progress, so this is kind of the first time I've brought it out in public. The hybrids tend to match one of the parents, and so this is true. This is only two of the more abundant crosses. But it seems like in each case where we have a substantial sample size of both parents and hybrids, we have a scenario where the counts on the hybrids are more closely matched to one of the parental species than the other. Another thing to look at here is that there's a bunch of variation in the lateral line scale count for the parental species too. We'll be digging back through that this winter and seeing if we think that it's real, if it's observer. We're going to have a second observer go through all these as well. But it appears actually that, you know, we miss the hybrids for good reason, is that the hybrids look like their parents. And so we'll be doing some more analysis of this and digging a little bit deeper as we go. And so this pattern is pretty clear for some traits, but it's less apparent for other traits. And so this is a little bit less of a giveaway. This is the trait of anal fin ray count, so the little cartilaginous bits through the fin. This is less clear, but it looks like in this case there's not much differentiation between the parental species either. So this is a hard thing about this clade of fish that all look roughly, that's too broad a generalization. A lot of them look quite similar. Western black-nosed dace doesn't really look like the others very much. But so this is something we'd like to do a lot more of is, you know, combining our genomic data with phenotypic data and trying to understand what hybrids look like, which is a key to understanding things like the ecological function of hybrids down the road. So that's a major goal for my lab going forward is to try and integrate the ecology and the evolution of hybrids in a satisfying way. And so to wrap up for today, we did find extensive hybridization among these fish species. The hybrids do resemble the parental species to a large extent in our data set, but the parents are also variable. And so something we've wondered about when thinking about the identity of these fish species is whether the original meristic counts for some things might have been unknowingly developed with hybrids in the mix. And so that all goes back to our question of, has this hybridization scenario been going on for a long time, or is this a new thing? I don't have a good answer for you, but we're continuing work on these species in my lab now. I'm currently at Northern Michigan University, and so we're extending this project or continuing this project in a new setting with different disturbances. So where we worked previously, we had agriculture and urbanization. Here we have mining and logging. We're going to establish sites this fall to continue work on these species up here. Okay, so I'd like to thank the many people at multiple universities who have helped with this work, the many funding sources, the computing support, and then I welcome any questions you have. Thanks.
Thanks very much. So I have a question. Do you think it is feasible or are there any opportunities to use museum specimens to address this big question of whether this is due to recent disturbance or is it kind of, evolutionarily old pattern?
I would love to use museum specimens. A really tricky thing we run up against with fish, though, is that many of our museum specimens are stored in formalin, and our methods for getting good DNA out of formalin preserved specimens are still not great. So there are some cases where they're ethanol stored specimens from prior to the common use of formalin. But yeah, there are other things we can do. There are statistical things we can do to try and understand whether this is archaic introgression or more contemporary stuff too.
Great. It looks like Ulrich Hegel has a question. If you want to go ahead and unmute.
Solange had written a question first in the
sorry.
Q&A. That's right. So what molecular DNA methods and techniques / approach did you use?
Right. Yeah, I kind of glossed over that. So we used a genotyping by sequencing approach. It's quite similar to a ddRAD or something like that, where we used restriction enzymes to chop up the DNA in predictable ways, and then we aligned to our reference genome, and then we identified about 10,000 SNPs from that data. Does that get at your question? Thanks.
Yeah, Ulrich, if you want to ask your question.
So my question would be, do you think that let's say the ease of hybridization that fish have compared to other vertebrates plays a role in the fact that fish have so many more different species than the other vertebrate groups?
This is $1 million question, and I love that question because I really part of the reason I love studying fish is because hybridization is all tangled up with with diversification of fishes. I can't answer that question because it's a huge one, but I think quite possibly that the ease of hybridization has led to evolutionary novelty in some clades.
Well, I think it's definitely not exclusive to fishes. I believe
Yeah.
I work mostly with insects, and I've had the
Hmm.
sneaking suspicion for a while. There's always these weird species that are found like once every 100 years or
Yes.
so, where,
Yes.
yeah, could be hybrids. Who knows?
Yes. I mean, there are clades of fish that arose from an allopolyploidy event 50 million years ago. So they're all historically hybrid tetraploids for
Hmm,
a little bit there.
okay.
Plants have a lot of this too. Fish are the plants of the animal world, is what I tell my students.
Yes, that's very fitting. Thank you.
Okay, we'll take, I guess, one more question. Sweet Ting, if you want to ask your question real quick.
There's a question in the Q&A. Is
Oh,
there a
again,
difference?
I'm sorry.
That's right. Is there a difference in the parents' effects on morphology of different genders?
Oh, okay. So these fish do not have sexual dimorphism for most of the year, so we actually don't know the sex of the fish we sampled. But that is, again, something we'd like to be able to do better in the future with really, I'm going in on all in on working on populations very close to my university right now to try and sample them in spawning period when there is sexual dimorphism, when so I can tell whether they're male or female. Yes, I would love to know this.
Okay, we've reached the end of our session. Just want to thank Rachel and all the participants and everyone for coming. And hope you all enjoy the rest of the conference.
Thanks, Kyle. And thanks, Rachel.